Hi, my name is Kate Martin, and I do research within the Weinman lab on campus. And today I will be presenting on applying line and groove patterns to nanofiltration membranes. First, I'd like to give a little background on nanofiltration membranes. There are four main types of pressure-driven membrane processes: microfiltration, ultrafiltration, nanofiltration, and reverse osmosis. Nanofiltration membranes are what are known as thin film composite membranes, which means that they filter out divalent ions, bacteria, viruses, and suspended particles. Nanofiltration membranes require a lower operating pressure than reverse osmosis membranes, making them a less expensive option for the partial desalination of water or water softening. There are three layers of nanofiltration membranes, with the top layer being the polyamide active layer, which is commonly synthesized using piperazine and trimethyl chloride. This layer is responsible for the rejection of divalent ions that nanofiltration membranes have. The next layer is the polysulfone, or the polysulfone support layer, and then the bottom layer is the polyester or polypropylene fabric. In our lab, we use a membrane that has polypropylene fabric. One major issue with nanofiltration membranes is fouling on the surface of the membrane. Fouling is buildup of solute on the membrane surface, which blocks the pores of the membrane, leading to a decrease of permeance and rejection of the membrane over time. The only way to clean a membrane that has fouling is to utilize harsh chemicals, which can damage the membrane and be quite costly. Line and groove patterns have been seen on the scales of sharks to decrease fouling on the surface of sharks, of foulants like algae sticking to the surface, allowing sharks to move more hydrodynamically through the water as they swim, which is why we are studying the effects of patterns on fouling by applying them to nanofiltration membranes, because patterns are thought to create areas of turbulence or localized mixing on the membrane surface, preventing the sticking of foulants. Specifically, our project is looking at the relationship between foulant size and pattern size and its influence on membrane fouling. We'll be looking at pattern sizes that are smaller than, larger than, and equal to the foulant size, which are 300 nanometer, 10 micron, and 50 micron pattern sizes. To pattern our membranes, we utilize stamps, which the 300 nanometer stamp we purchase, and the 10 micron and 50 micron stamps we fabricate using photolithography. We stamp a membrane, a PS20 membrane, which is the bottom two layers of the nanofiltration membrane. It's an ultrafiltration membrane that we purchase. It does not have the polyamide active layers. So we use thermal embossing to stamp the pattern onto the membrane surface, and then we'll go in and synthesize the polyamide active layer on top so that the pattern is under the polyamide active layer. And the patterns are shown in the top image here, the stamps. To synthesize the polyamide active layer, first we soak the PS20 membrane in deionized water overnight, and then we tape it onto a glass plate and pour on an aqueous solution of piperazine and deionized water. This is allowed to sit for 2 minutes. We then pour it off, and we use an air knife to completely remove the aqueous solution. Then we pour on the organic solution, which is trimethyl chloride and dodecane. This is left on for 1 minute for the non-pattern and 300 nanometer pattern membranes, and 1 minute and 30 seconds for the 10 micron and 50 micron pattern membranes. The organic solution is then poured off, and the membrane is rinsed with hexane. Then the membrane is left in a water bath for 45 seconds, and then the membrane is removed and cut into the shape we utilize for testing. And this is SEM imaging of the membranes after the synthesis process. So the first image, image A, is actually a purchased nanofiltration membrane called NF270. This is what we compared our in-house synthesized membranes to. And then the rest of them, we synthesized the polyamide active layer on top, and you can clearly see the different pattern sizes in these images. Now onto how we tested the membranes. We utilized a dead-end cell with stirring. The membranes were tested at 150 psi, and we initially tested with DI water to test for pure water permeance, and then with a 2,000 parts per million sodium sulfate solution to test for salt rejection. We then did oil fouling tests with first testing with deionized water, and then we tested with a oil brine solution, which was 1,000 parts per million dodecane and 35,000 parts per million sodium chloride, which is similar in composition to the wastewater from fracking. Initially, we tested for the pure water permeance and the salt rejection of our in-house synthesized pattern membranes and non-pattern membranes, and then we compared that to the NF270 membrane. These membranes are known as pristine because they haven't been tested yet, so they haven't experienced fouling yet. And these tests demonstrated that the membranes we fabricated had a slightly lower pure water permeance than that of the NF270 purchase membrane, but we saw no significant statistical differences between our in-house synthesized membranes and the membrane that was purchased for the rejections. And another thing that we saw from this test was that for the synthesized membranes, we saw no statistical difference between the rejection based on the patterning or the pure water permeance. The patterning did not influence that for our synthesized membranes. Next, oil fouling tests were run. The membranes were tested with an oil solution feed, then manually cleaned, then they were tested with a DI water feed, and this was repeated 3 times. For each test, the water versus the flux versus the water throughput was measured to observe whether water flow was restricted by foulants, and this is shown by the graph to my right. From this graph, the rate of fouling for each membrane can be observed by the slope of the line in the oil sections. The more steeply the line declines, the higher the rate of fouling because you are getting restricted flux. Through this, you can see that the 300 nanometer and the 50 micron polyamide membrane had a decreased fouling rate, leading to the understanding that larger than and smaller than patterns do reduce the rate of fouling. Additionally, the water flux recovery can be seen by how the flux recovers before and after oil fouling, so after the manual cleaning, how the flux recovers when the water's being run. And we were able to see from this graph that there was no statistical difference for the flux recovery ratio, whether that be positive or negative, there's no statistical difference. Additionally, we observed the water contact angles before and after fouling. Contact angles for the NF270, the unpatterned pristine, and the 300 nanopattern membrane were all statistically different before and after fouling, showing an increase in hydrophobicity, which is likely due to oil on the membrane after the oil fouling tests. The contact angles for the 10 micron and 50 micron patterns were statistically not different, but that's likely due to a hybrid wetting regime on the patterns. So initially, when the contact angle is being taken before oil fouling, they're more hydrophobic just because the water doesn't fully sit within the pattern, so leading to higher contact angles before oil fouling. So there's no statistical difference between, statistically significant difference between the before and after oil fouling for the 10 and 50. Overall, the smaller than and larger than patterns reduce fouling rates. And in the future, we plan to utilize crossflow cells, which are commonly used within industry. And we used a dead end cell with stirring to mimic a crossflow cell for our tests. And so in the future, we plan to utilize crossflow cells. Thank you. Any questions? Michael.
So in the synthesis procedure for the polyamide layer, you mentioned that the organic solution was left on longer for the 50 and 10 micron membranes, and I was wondering why that was.
So for the 50 and 10 micron membranes, the pattern is larger. So we left the organic solution on for 30 more seconds because we were seeing that the polyamide active layer wasn't fully formed, so we wanted to ensure that it had enough time to fully polymerize over those grooves. So we left them on for 30 more seconds, and that seemed to be enough time to allow the polyamide active layer to fully polymerize over the 10 micron and 50 micron grooves.
Why did the membranes that your lab fabricated have a lower pure water permeance than the commercial membrane?
So with the synthesizing of the polyamide active layer, we think that we had a thicker polyamide active layer than the purchased NF270 membrane. So this led to a lower pure water permeance, but it didn't affect the rejection of the membranes.
So I just wanted to ask, why did you not use the cross-flow cells initially?
We didn't initially use the cross-flow cells because we didn't have access to them, but we now have access to a cross-flow cell with Dr. Esfahani's lab, and we will be repeating these tests with the cross-flow cells. But what we did use was a deadened cell with stirring, so that created movement over the membrane, which mimicked a cross-flow cell.
I had a question. I think it was slide 8, where you showed like the step heights of the different. I think, yeah, this slide. Yeah. So on the second, I guess, image of the step heights here, I see like the regular drop-offs, but then also at each peak, there's this like small divot that happens regularly. Was that an intentional part of like the stamp, or is that like an artifact of the stamping process? And does that have any like effect, I guess, on
Are
that?
you talking about the so
Like the image
10
under,
micron and then like below it?
yeah, the graph under the 10.
So when we were doing the profilometry, like sometimes the tip didn't get fully in the grooves, so that might be due to it, because it was too tiny to sometimes get fully within the grooves for the profilometry, so that's probably what you're saying. But also, it wasn't fully like, we wanted it to be 10 micron deep, 10 micron across. It wasn't always 10 micron deep with this stamping because we use thermal embossing, but it was pretty close.